Hi everyone! Today we are going to be looking at plant propagation. So this is kind of a preview of a larger course that we offer here at Rowan. I actually teach it. I'll be teaching it in next semester. So if you get interested in what you see in this lecture, sign up for the class coming up. So anyway, before we get into this, we are going to review plant genetics. So last lecture, we looked at plant genetics, and a lot of it was stuff that we had already covered, right? So we looked at genes, and again, they were segments of DNA that code for particular proteins. We talked about genotype, which is the genetic blueprint of an organism, and then the organism's phenotype, which are the observed characteristics, right? The actual traits that we see. And that phenotype is made up or determined by the genotype and the environment, right? And then we talked about, well, a gene is a segment of DNA, and that DNA is actually packaged into what we refer to as chromosomes. And most organisms have two copies of each of their chromosomes, right? We refer to this as 2n, with n being the number of copies. But, for instance, humans, we have 46 chromosomes, we have 23 individual chromosomes, and then a second copy of each, right? We get one copy from our mom, one copy from our dad. But sometimes organisms, particularly our plants, can be 3n, 4n, 5n, multiple copies of chromosomes, and we refer to this as polyploidy. Right? I think in the lecture I said that even ferns can be up to 400n, which is just ridiculous. So then, because we have two copies, for each of our genes, we will have potentially two different forms or versions, and we refer to them as alleles. Now, our alleles, for the most part, can be either dominant or recessive. And if they are dominant, it means that allele, it always expresses whatever trait it's coding for. I gave the example of flower color. So if purple flower color is dominant, if we have the allele for that purple flower color, our flowers are going to be purple. Recessive alleles are only expressed when they are combined with another recessive allele. Right? If white flowers is recessive, we would need two copies of this recessive allele for us to actually see, or for the white flowers to be expressed, for those flowers to be white. Now, when we are looking at gene combinations or genotypes of a trait, we can say that homozygous is when they have the same alleles, and heterozygous if there are different alleles, right? So, for instance, we're going to talk about flower color again. If P, capital P, is for purple flowers, lowercase p is for white, right? We could have an instance of capital P, that is homozygous. Both of those alleles are the same. That would be homozygous dominant, we would typically say. But you can also have the case of lowercase p, that's also homozygous. Both of those alleles are the same. In this case, they are both coding for white flowers, and we would see white flowers here. But besides the homozygous gene combination or genotype, you can also have what's referred to as heterozygous, and that's where for your gene you have different alleles. So you would have in this instance, you would have a big P and a little p. So one allele that is coding for purple flowers and one that's coding for white flowers. And if you recall, since the purple gene is dominant, these would be purple flowers. Right? Okay. The other important thing is sometimes we have what is referred to as hybrid advantage. And that is the case where our hybrids, which are typically our heterozygous organisms, right? So you have an inbred line. We see this with our corn. We have an inbred line here that would have all, in this example, P. capital P, and then we'd have another line here, and then we would cross them, and it would give us these heterozygotes. This heterozygote would have advantageous characteristics over these other lines, right? And that could be like increased corn yield in particular. Okay, lastly, we're going to look at Punnett squares. So a good way of keeping track if we're making crosses, if we're breeding plants together and keeping track of their genetics is by doing a Punnett square. And they can get more complicated. You can only use them up to a point, but if you're looking at single genes like we're looking at here, what you do is you take your genotype of your adult. Say we're crossing a white flower, so we're going to know that genotype, little p, and we're going to cross it with, we know that it is a heterozygous plant. So big P, little p. I probably should have taken easier ones to determine between capital and lowercase. But you line up each allele for each plant, and then you bring them down into each box. So you bring your big P across here, and your little p down, big P across, little p. Whoops, hit the wrong button. Little p, little p, little p, little p. Right? So if we made a cross of these plants, our offspring should be about, we have two heterozygotes, so big P, little p's, and two little p's. We can just kind of say that's 1-to-1. So you should have about 50% of your offspring would be heterozygous, and 50% would be homozygous recessive. So these would be purple flowers, and these would be white. Okay, that is good. Okay, so plant propagation, in essence, what it is creating new plants, right? You could say the process of increasing plant numbers, but it's just making new plants. Now, successful propagation involves transmitting all of the desirable characteristics of the original plant or plants to all progeny, right? We have adapted or modified largely throughout history through selective breeding, ancestral varieties of crops into modern day crops that we know now, right? And we've tweaked all their traits. So what we want to do is, as we create, and we've made those traits beneficial for us, whether it is, higher yields or increased fruit size or increased sugar content, etc. Right? So when we propagate, when we are going to create new plants, we want those new plants to have those same traits as the parent plants, right? They are desirable traits, and we want to make sure that the new plants have those same traits. So that's one of the big considerations that we take when we are doing propagation. And that consideration is going to influence some of the methods that we use, whether we are propagating by seed or whether we are propagating vegetatively, as we're going to look in this lecture. So that's an important point. Some other just kind of definitions. A cultivar, you may see this, you may have heard this term, stands for cultivated variety. So these are plants that have been selected for those traits that I talked about, and those traits are maintained. So we have different cultivars, or sometimes you will just hear variety of plants of select species that have particular traits, and we want those traits to persist through time. So, as I mentioned, we have two methods of propagation, two main methods. We have seed propagation and vegetative propagation. And there are important distinctions between those. One of the first being that seed propagation is a method of sexual reproduction, whereas vegetative propagation is asexual reproduction. So we're going to start with seed propagation, and listen, we have gone through this process of pollination and fertilization a number of times, so I don't need to get into it. But what I will talk about is this idea of sexual reproduction as a whole, because I think it's interesting, right? if you think about it from an evolutionary standpoint, why does sexual reproduction exist? Why did it evolve? Right? It is, if the goal of an organism or of life is to create more life or to pass on your genes, asexual reproduction seems a lot easier. Right? You can just make a little bud of yourself and, oh, there you go, you have a offspring. Sexual reproduction requires a lot more. It's a lot more difficult process, right? You have to find a mate. Often that involves, especially if we're talking about animals, you have to travel to find your mate. There's often competition between individuals to have access to mating rights. There are whole hosts of characteristics that have evolved just to attract a mate, right? You think of birds. Birds have evolved these elaborate colors, especially the males. That's why the males are so elaborately colored. That is to attract the females. And those colors, birds such as the bird of paradise has a huge long tail. These bright elaborate colors, while they attract a female, they are detrimental to the male bird itself, right? Those bright colors also attract predators. And this is referred to the handicap principle. It's why they think these bright colors are so attractive to the female. If this male bird can be so brightly colored and can have a big ass long tail, that is going to make him easier, or presumably easier, to see and catch and by predators. And if he's still alive, he must have really good genes. So I may want to mate with him because my offspring are then going to have really good genes, and there's a better chance that they're going to survive and produce more offspring. So there's a lot of downsides or costs of sexual reproduction, even with our plants, right? There's costs that go into the development of flowers, to the mechanisms that are going to facilitate that. Our plants put a lot of effort into, especially if they are pollinated by insects, to attract those insects. And these are the energy that goes into, whether it's producing nectaries or flowers or all of that stuff, could be put into just growing a plant or creating a bud that would, or some sort of offspring or vegetated offspring. So there must be a really good reason why, or really good benefits to sexual reproduction to outweigh all those costs. And what scientists think, what the kind of prevailing theory is the importance of sexual reproduction is a way to prevent parasites and disease, which is really, really interesting. So the big benefit of sexual reproduction, and I'll think in the next couple slides we'll talk a little bit about it, but the big benefit of sexual reproduction is that it mixes up gene combinations. We saw this a bit with the Punnett squares, and when I talked about the hybrid vigor and why you can't cross two hybrid plants and expect to have the same phenotypes or genotypes, because that process of sexual selection or sexual reproduction mixes up gene combinations. So your parents you yourself are a combination of the genes of your parents, right? You're not exact copies. Well, that mixing up of genes is really important in preventing parasites or preventing certain diseases, right? Parasites and certain other viruses or bacteria, etc., have pathways that they enter into your body, and particularly your cells, right? So there are certain pathways that they use. And those pathways are dependent on like characteristics of your cells, for example, right? So there may be a certain protein on your cell that this parasite is going to be able to use to access your cell and do all the things that it's going to do. Well, that protein, that is the access way for that parasite, is determined by a gene. And if you mix up the genes, it may be that your offspring won't have that exact protein, right? So your offspring will be safe from that parasite. It's a really kind of neat way of thinking about it. And there's evidence of this, right? There are snails. I believe this work was done out in California. And double check me on the internet. You can look this up because I don't know if I have the specifics right. But I believe it goes like this. So there are snails, aquatic snails that live in these pools out in somewhere out west. Like I said, I think it's California. Right? And our snails can switch from sexual reproduction or asexual reproduction, right? They can either reproduce sexually or they could reproduce asexually. Now, in these pools where there are no parasites that prey upon the snails, these snails will by far reproduce asexually, right? There's no parasites there, they're just creating little buds of themselves, little clones of themselves. Where there are parasites, in pools where there are parasites, these snails will instead reproduce sexually, right? So that, with the idea being that their offspring, by reproducing sexually, like I said, that mixing of gene combinations will help protect their offspring from those parasites. So one kind of cool example that provides evidence to this theory. It's called the Red Queen hypothesis, if you guys are interested. So it was a little bit of an aside, but I like sharing those types of stories about evolution because I think it may provide a greater appreciation for life in the natural world and how unique and complex it could be. So, anyway, a little bit of an aside on sexual reproduction. But when we're talking about seed propagation, it is that process of sexual reproduction. Now, we will have what are referred to as lines. Now, our lines are plants that reproduce true to seed. So your progeny, so you have your adults and they're gonna reproduce through seed, that's that sexual reproduction. But the progeny are gonna share the same characteristics as the adults or the original plants. And this is related to genetics, as I talked about, right? Our line has to be homozygous, or largely homozygous. And if you recall from our genetics, homozygous is where you have the same alleles for all of your traits, right? So if we have a little Punnett square up here, if we're looking at just one single trait, both parents, one parent's up here, the other parent's up here, both of them are homozygous for this trait. So they have the same alleles everywhere. So their resulting offspring are going to have the exact same genotype, right? If you have a cross of parents that are more heterozygous for a number of traits, the resulting offspring are going to vary in their genotypes, right? So you wouldn't have a line in a very heterozygous, in a population of plants that are very heterozygous, right? Their offspring will most likely not reproduce true to seed, at least most of the offspring, or a portion of them, right? And I already talked about that when I talked about hybrid seed, right? With our hybrid seed, only about 50% of the offspring are going to have the same genotype, and then thus same phenotype physical characteristics. So, in the United States, there are seed certification programs that essentially have been developed to protect and maintain the genetic quality of cultivars or varieties. And within these programs, if you get into it, there are 4 classes of seed, right? There are breeder seed that's produced in small amounts, that's controlled by the plant breeder, and that is used to produce foundation seed. That foundation seed gets multiplied from the breeder seed, and that produces more foundation seed, and then it's used, and then it's controlled by the seed stock organization. Then we have what's referred to as registered seed, which can be either from the breeder or foundation seed, and this is the seed source that growers of certified seed use. And then you have finally certified seed, which is the seed that is available in large quantities that's sold to farmers or the general public, right? So it's just ways of maintaining the quality of seed that we have available. Really, the seed that you are going to be buying would be certified seed here that is produced from registered seed, which itself is produced from foundation seed or possibly breeder seed. Now, For a seed to germinate, the embryo must be alive. And the time that the embryo remains alive will vary within species. For example, willows, maples, and elms, the embryo has a short life in that seed, whereas some other species, particularly your hard-seeded legumes, such as clover, the embryo will survive for years. Right, and this is related to the different seed characteristics, which we won't get into too much here. Now, if you're going to be planting a whole ton of seed, you may want to test a couple of seeds before planting them to ensure that the majority are viable. And there's a couple ways you can do this. You can cut the seed in half to look at the embryo, make sure you can see a visible, healthy-looking embryo. You can float some seeds in water, and often the floaters are empty seeds, right? So you would discard them, right? They don't want to have a viable embryo. You can x-ray your seeds. There's a tetrazolium test, so you can essentially cut your seeds in half, apply this tetrazolium chloride, which will turn red when it's exposed to respiring living tissue. So you can see that if it turns red, you have a viable embryo still. Or you can actually just do a germination test. You can plant a couple seeds, count how many germinate. You don't need to necessarily plant them in soil. You can just use wet paper towels like we did with our pea seeds or in our pocket seed lab. So seed dormancy. I mentioned seed dormancy in the past, right? Seed dormancy is essentially suspended animation for our seeds, right? As we learned, for germination to occur, you need three main things: water, the right temperature, and oxygen. Sometimes some other factors, like I mentioned about cold, fire, smoke, etc. But if you have those things, those are the critical things for germination. But sometimes you may have those three things, and your seed will not germinate, and that is because it is in this dormancy condition. And nearly all of our seeds start off as dormant. Some more so than others, and it's believed that we have bred, most of our crop seeds their dormancy condition isn't as extreme as some others, and it's likely that we bred that into them, right? So they wouldn't be as dormant as some of these other seeds. But seed dormancy, there are two main types. There's exogenous dormancy and endogenous dormancy. And we won't go too deep into them. I'm going to give you the biggest examples of each, right? So when we're talking about exogenous dormancy, that is dormancy related to factors other than the embryo. And the most common one is the seed coat, right? In this instance, the seed coat is such that it prevents water and oxygen from reaching the embryo. And we see this in alfalfa, clover, pine, birch, right? It has this hard, impenetrable seed coat that is preventing those two important things, water and oxygen, from reaching the embryo, and thus that's going to prevent germination from occurring. So it's only after that seed coat gets worn down that germination will occur. And that seed coat will get worn down naturally from weathering, right, being exposed to the environment, or the action of microorganisms that start to break it down, or possibly passing through the digestive tract of an animal. Now, for us, if we're propagating, we have a couple different ways to actually break that seed coat dormancy. We have scarification, where you can take, like some sandpaper, rub it against the seed to start breaking up that seed coat. There is heat treatment, so you can boil or expose your seeds to hot water, and that's going to start breaking down that seed coat. But there's also something called acid scarification, where you can actually soak your seeds in sulfuric acid for a period of time to break down that seed coat. Now, that's exogenous dormancy, and that seed coat dormancy is a type of that. The remaining is endogenous dormancy, and it's stuff related to the embryo, and we're going to refer to it here as embryo dormancy. And like I said, it's some physiological aspect of the embryo that keeps it dormant. And it's related to hormone levels, as we'll see there on the bottom, abscisic acid versus gibberellins, etc. They think we don't really know the the mechanism entirely figured out, right? Um, but for our embryo dormancy, to alleviate that dormancy, for most of them, you're going to require some period of stratification that we've learned about, right? And that is that exposure to cold temperatures. And different species will require a different length of exposure, right? And doing that, as you see down here, doing that affects these hormone levels that we have found that out, right? So through the stratification process, abscisic acid decreases, and if you remember, abscisic acid is that hormone that maintains dormancy, and gibberellins increases, and gibberellins is the hormone that is going to promote germination. So that is kind of the overview of seed propagation. And now we're going to get into vegetative propagation. So here, instead of sexual reproduction and utilizing growing new plants from seed, we are going to take vegetative tissues, and from those vegetative tissues, which could include the stem or leaf or the root, we're going to use that to develop new plants. And the basis of this all relies upon the ability of plants to form adventitious shoots or roots, or both. And what they are essentially shoots or roots that develop from any place other than the main system, right? So if we're talking about adventitious shoots, we're talking about shoots that develop from any place on the plant besides the main shoot terminals or in the axis of the leaves, right? So anywhere besides this main shoot area. So if we took a leaf and we grew a stem from that leaf, that's an example of an adventitious shoot. Adventitious roots are roots that develop from any place besides the main root system, right? Again, if we're looking at a leaf and we have roots that are going to start developing from that leaf, or roots that develop from a cut stem, those would be adventitious roots. So, like I said, extremely important. The basis of all vegetative propagation is the ability of many plants to form either adventitious shoots or roots. Now, like I said, vegetative propagation is an asexual type of propagation. You are creating clones. All of your plants are going to have the exact same genotype as your mother plant. Right, and this could be particularly adventitious for plants. I struggled with that word. For plants that are highly heterozygous, right? So they have a lot of different allele combinations, right? So they won't breed true, but if you take a cutting from it, it's going to have that exact same genotype. It's going to have those exact same traits as your mother plant. So where do new cultivated clones originate from? Right? Where do we get these new varieties in vegetative propagation? Really, a lot of our fruit species are all propagated this way, right? Through cuttings or layering. So where do we get these new varieties? Well, it's two ways. Sometimes we have seedling plants that are discovered. So we'll just discover a wild plant that has these really cool characteristics, and then we're going to propagate them vegetatively to maintain those characteristics. As an example, Golden Delicious apples were originally discovered by A.H. Mullins on his West Virginian farm in the early 1900s. So he just happened upon Golden Delicious apples, and then we've maintained that line, that variety, through the decades by propagating vegetatively. Or sometimes mutations will occur in a plant, and those mutations will have a desirable effect, and then we can keep those traits, that new mutation going by creating clones, right? And the example I gave there is the pink-fleshed ruby grapefruit, originated in 1829 as a mutated branch on a Thompson's pink grapefruit, right? So on this branch of this Thompson's pink grapefruit, we had a mutation that occurred that resulted in a ruby-fleshed or pink-fleshed grapefruit, and then we maintained this characteristic by propagating that off of that branch. Another kind of interesting thing that relates to propagating vegetatively are chimeras. So a chimera is a plant composed of cells with multiple genotypes growing in the same tissue. So you may have a leaf or a root or a stem or shoot that consists of different genotypes. Typically, your plant, right? They all of, as we learned in the genetics, all of the body cells, the vegetative cells, should have the same genotype. But in certain cases, like in a chimera, there is a mutation that occurs where you will have different genotypes in the same tissue. And that can result in some really unique traits, right? These variegated leaves that we see sometimes can be the result of a chimera, or thornlessness in blackberries is a result of a chimera. So what's happening, and we won't get too in-depth with this, there's different types of chimeras, and it gets a little complicated. But if you look at your shoot apical meristem, remember that's the spot, the tissue that is actively dividing tissue. It creates all the other tissues in your shoot. It has 3 layers, and those layers develop into different tissues. So this L1 layer is going to develop into the epidermis, while this L2 and these L3 layers develop into the other types of tissues that we talked about, whether it is cortex or xylem, phloem. Hold on, my dog's jumping down. There you go. Xylem, phloem tissue, etc. So depending on where a mutation occurs in these tissues will result in a different type of chimera. Most importantly, if it develops in this L1 area, that mutation, and if it is called what we refer to as a paraclinal chimera, that mutation is going to occur throughout the epidermis, and it's going to result in certain characteristics, like thornless blackberries, right? And what I want you to just get from this is that this is the only one in which we can propagate the traits continuously, right? So we need this mutation to occur throughout the entire layer, and if it does, then we can propagate that vegetatively. What we can't do is, if we have these thornless blackberries, like I said, it's a result of this mutation that occurred here. If you try to propagate them through seed, they are going to develop thorns because the genetics of the remaining tissues would be for the thorned variety. It's just this random mutation that occurred in the epidermis that results in thornlessness. So we have kind of 4 main vegetative propagation methods. We can do cuttings, we can do grafting, we can do layering, and then there is now micropropagation. So in cuttings, you're taking either a piece of stem or a piece of root, or in some cases a leaf, and from that you are developing the missing parts to create a new plant. With grafting, you are joining parts of plants together so they unite and then grow as one plant. With layering, what you're doing is very similar to cuttings, but instead of cutting that entire stem off and then planting it and having new roots grow, you keep the stem attached to the plant system, right? You modify it, you scrape away essentially all the tissues except the xylem. So you maintain water flow to your what's going to be your cut part, and in this process, what is going to be your new cutting, you set it up so it will start growing roots while it's still attached to the main plant, and then once it grows roots, you cut it off and you plant it. It's beneficial because one of the issues that you get with cuttings is water loss, right? Especially if you're cutting off a stem, our plant is taking in water through the roots, and this stem initially has no roots until new adventitious roots develop. So it is susceptible to water loss at that point. By layering, you're already growing roots, so you're helping to alleviate that issue. And then micropropagation is done more in a laboratory setting where you use very, very small pieces of plant tissue grown on a media, and you develop the whole plant from just small pieces of plant tissue. So again, we'll just look real quick at these. We're not going to go crazy in depth. But different types of plant cuttings. You can do stem cuttings, leaf cuttings. You can do a leaf with the petiole, leaf sections even, and then root cuttings. Right? And all of these could be used depending on the different type of plant to propagate, make new plants. There are some factors that influence cuttings, the source of the cutting material, especially if we're talking about woody plants. You may want to use, for many of them, juvenile plants. Right when they go through that phase change and they become mature, they often can't be used. You can't take cuttings from them for some of our plants. The time of year that you take cuttings. So hardwood cuttings, if you're taking hardwood cuttings, they're often going to root best if you gather the material in late winter. Right? You store it and then you're going to plant it in the spring. Softwood cuttings, you're going to take that material in the early spring, right, and cut them and plant them. For herbaceous cuttings, you basically can take them any time of year. Idolation is something you can do to improve the initiation of adventitious roots. So idolation is essentially where you're growing the plant, or at least a section of the stem, in darkness. If you know you're going to take a cutting, you may wrap some tape around a certain section, keep it from getting light. That creates this idolation, and it essentially makes it more likely for adventitious roots to grow from that section. So you would keep it covered, take your cutting, uncover it, plant it, and it's going to make it more likely for roots to grow. As I mentioned in the hormone lecture, auxins are plant hormones that help stimulate adventitious root growth. You can buy pre-mixed auxin concentrations. You can make your own. Generally, you want a concentration of either 1,000 to 10,000 parts per million, 10,000 parts per million being pretty high. But you would take your cutting, dip it in the auxin, and then plant it. Like I said, that drying out, water loss is a big issue with your cuttings, so you want to make sure that they're kept moist to prevent them from dehydrating. And then applying some bottom heat will also help facilitate rooting. Right, and this is stuff that that we can investigate in plant propagation this year, which will be pretty fun. Then we have grafting. So grafting is essentially combining two different plants together. You're making a Frankenstein plant. And there's some different reasons of why you want to do it. One of the big ones is you can combine the benefits of both types of plants, right? So you can get a very good rootstock, and maybe that plant doesn't produce really great fruits, but it has this awesome rootstock. And you can cut off the top part of that plant and attach a variety or part of a plant that produces really great fruits. And now you've got the benefits of both. Right? So the scion is the part of the graft that's going to become the upper part of the plant, typically the shoot system. And the rootstock is the part of the graft that's going to be the root system, or the bottom part of the plant. And there's different types of grafts that you can do. I'm going to show you in our lab video a couple different examples of the types of grafts. But again, if you take plant propagation, we will look at these a little more thoroughly. And like I said, you can combine characteristics of plants. So one of the things that is common in orchards is that they will graft varieties of apples onto dwarfed rootstock. Right, so your resulting tree is going to be dwarfed. You're allowed by doing this. You can typically fit more trees in an orchard. It makes it way easier for you to care and harvest the apples. In this case, I say apples because it's common in apple trees, but it's also other orchard trees as well. Layering, again, like I said, very similar to cuttings, and there's different types of layering that can be done. In all instances, you are maintaining your propagule, or what is going to be your new plant. You are keeping it connected to the mother plant until typically roots grow, and then you sever it, right? So tip layering, you may take a stem off of this plant, plant it into the ground. It grows, in this case, it grows a new root and shoot system, and then you sever that propagule, and now you have a new plant. Simple layering, again, maybe more common. You bend it through, plant a portion of it, that's going to grow new roots, and then you can cut that off. Air layering, we have done before in propagation. They did it in previous classes. I did it this year on a blueberry plant that I have outside. Here, like I mentioned before, you're stripping the majority of the stem section, pack it with material. It could just be potting soil. Wrap that up and give it time and let new roots grow, and then you sever that connection. And then lastly, there are some natural propagation methods, right? So there are a number of plants that have evolved natural methods to propagate asexually. For example, our strawberry here produces stolons, which are these horizontal stems, right, that grow, and from those stems at nodes we will get new root and shoot systems. We have rhizomes, which are just the underground version of those, right? Stolons are above ground typically. Rhizomes are underground horizontal stems that at nodes will produce new shoot and roots, right? These rhizomes, if you remember way back in the beginning of the class when I did that edible plant walk, here's a cattail, here's that cattail rhizome. The tip of that rhizome here, or new rhizomes, are what I refer to as cattail laterals. They are really tasty to eat, right? So you can dig through there and find some of these white rhizomes, new rhizomes. You can harvest those and they're pretty tasty. Number of plants produce rhizomes, cattails, banana, ginger, right? All produce rhizomes. Suckers are adventitious shoots produced from horizontal roots, right? So with stolons and rhizomes, they are horizontal stems. Suckers are shoots that are produced from horizontal roots, and these occur in blackberries, for example. So here are roots that are horizontal, and from these roots you can have a new plant that starts developing, and we refer to as a sucker. We also have some other types of natural propagation methods. Crowns are the junction between the root and the shoot, so where your root and your shoot meets in your perennial herbaceous plants or monocots. And your crown will increase each year as your new shoots arise. So remember, our herbaceous perennials live more than 2 growing seasons, right? But typically the vegetative section is going to die off each winter. And what persists is the root system and this crown that you see right here. And that is going to increase each year as you get new shoots. This becomes storage material throughout the winter and then provides energy for the new plants, the new shoot system to develop. Well, these crowns can be separated and transplanted, right? We can see this with asparagus. I do a lot of work along beaches and American beach grass. We separate the crowns of those when we're trying to propagate new beach grass. We also have some bulbs, corms, and tubers, which are specialized stems that act as storage organs, right? And they create little offsets or bulbs that can be broken off and planted as new plants, right? These little bulbs that can be produced here. On a corm, these things are smaller things called cormels, right? That can be detached and planted. Tubers, we get new shoots and roots that can form from the eyes of your tubers, right, of your potatoes. All natural propagation methods. Besides our tubers, which are actually stems, we have tuberous roots, right? Think of our sweet potato. And again, this is root tissue that is storing nutrients. But if you plant this, it will grow adventitious shoots for your root, and you will start developing new roots as well. So examples are sweet potato. And lastly, the last type of vegetative propagation is micropropagation. And like I said, this is typically done in a lab where we take small pieces of a plant and we are going to grow it on a nutrient media. And by that nutrient media is going to contain all the necessary characteristics, all the necessary things that you need for a new plant to grow, including different hormones. Right? We do this, and this can be done on a number of plants. I went to before the start of the pandemic in January, we did a family trip to Disney. And in Epcot, they have a huge greenhouse that is all hydroponics. I did a tour of the greenhouse, like a nerd, and within there, they have a section that they were doing micro propagation, right? So they were propagating, I don't remember what they were actually growing. It might have been any number of the plants that they were growing in there. But they had a whole lab section essentially set up where they were propagating in this way. So, something interesting. I don't get too into it here. I think in plant propagation, we'll talk a little bit more about it. We may go more in depth this upcoming semester for the class. All right, so that is it. That's a little preview of plant propagation. That is it for now. I will see you guys later.